Can everybody hear me okay? I think you can. All right, it's great to be here today and have the chance to share some of my work with you. So being that we're here at the NITMB, I thought I would just start by saying how enthusiastic I am that the center now exists. And so I went to the NITMB website when I was making my slides to kind of like see how they articulated the core goals. And as we're probably all familiar, the idea is that the NITMB aims to integrate mathematics and biology in order to both transform biological research and to inspire new mathematical discoveries. And if you read a little bit further, there's a quote, research supported by NITMB is structured so that theorists and experiments collaborate on experimental design and data analysis as well as modeling. NITMB also supports the development of new mathematics inspired by biology. Now, what I want to point out is I endorse this fully, and that first sentence, I think, is extremely common in centers involving the intersection of biology and experiment. That pretty much everybody recognizes that you need to have experimentalists and theorists working together to kind of best do biology. But NITMB takes it a step further, and they say we also want to get new mathematics inspired by biology, and I also think this is really critical. And so I think it's really valuable that the Simons Foundation and NSF have stepped up to kind of like recognize this fact and invest in projects that do this. So my core department is neurobiology, and my main goal is to understand neuroscience. And so I want to start by just introducing you to kind of the state of experimental neuroscience. I'm a theoretician. That is where I'm going to draw some initial inspiration. So we're in this very exciting era of very large and complex data. So what you're seeing on the right is a movie of whole brain activity of the larval zebrafish brain. In this particular case here, the fish is kind of just freely exploring its environment in virtual reality. And you can see lots of flickering activity that would represent a fluorescent readout of a calcium indicator, which is in turn indicating neural activity. And one of the things that's really amazing about larval zebrafish is you can do this at the scale of the entire brain and at the resolution of single neurons. At the same time, efforts are reconstructing the synapse resolution circuitry of the entire brain using electron microscopy. So here what you have is a detailed wiring diagram of the network that underlies these dynamics that you can observe functionally. And I think that there's been a long-standing central aim in neuroscience to kind of understand the quantitative link between what is the structure of the function in the brain and is now becoming empirical. And what's exciting about this for me also as a modeler is that this fundamentally requires neural network models of some kind, because you need a mathematical description that is going to relate the structure that you can observe experimentally now, the dynamics that you can observe experimentally now, through some mathematical model that ties it together in some form. And I don't want you to worry about the details of this model, because I'm going to be going through aspects of this and many other models as I go through, but I just want to kind of give you the sense that we're in this era where we can actually record both the dynamics and the structure of a neural network, and modeling is critical now to make sense of what this means. Now, I want to just point out, I think, a very profound issue, which is that even in very, very small circuits we've been able to characterize comprehensively for a long time, it's been known that there's a difficulty in terms of bridging between the structure and the function of a network. So here's an example from Yves Marder's lab that I find very inspiring, where we're just looking at the rhythmic patterns generated by 3 types of neurons in the crab stomatic gastric ganglion. And you can see that these 3 types of neurons are oscillating, and they have this sequential pattern of activity. Now, the challenge is not to devise a mathematical model that can do it. So this is an example from Yves Marder's work, where she kind of built a detailed biophysical model of this circuit. There are parameters involving the membrane conductance of how neurons respond to their input. There are parameters describing the synaptic conductances, how they communicate with each other. And actually, I presented that as data, but that's actually a model. It's a model that basically looks exactly like the data. The problem is you can come up with another model that looks exactly like the data, and the parameters look very different. So it's not a matter of just finding a network that can account for some dynamical property. That bar is too low. So the approach that I like to think of Yves Marder having taken, and I'm going to take throughout this talk, is what I call ensemble modeling. And the concept here is that rather than building and analyzing single models, we're going to try to characterize and analyze the entire ensemble of models that could work in order to kind of make our biological inferences and try to understand how the system works. And as a quick public service announcement, this notion of ensemble modeling is not the same as the ensemble model we just heard from Becca Willett. in the sense that both of them do kind of look over a class of models, but here I'm not going to be averaging them or anything like that to create a single model. I'm going to be analyzing the space of models itself. Now, if I want to analyze the space of models itself, Eve Martin and colleagues have done that for some of these simple biophysical models by just doing brute force numerical exploration of the parameter spaces. But that's really not feasible if you want to understand neural networks on the scale that I introduced them before, where you're looking at the entire brain, where there's, in the case of the zebrafish, there's 100,000 neurons. There's many, 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 many things that are unknown. It's just not feasible to hope that we can build sort of a detailed biophysical model. And throughout this talk, I'm going to be using kind of an abstracted version of neural network dynamics that is commonly used among computational neuroscientists called firing rate models. And so I want to kind of quickly explain what that is. And so the idea would be that in a real biological neuron, if you inject some current to that neuron, the neuron responds by firing action potentials. And for example, you could quantify what would be the firing rate of that neuron as a function of the injected current. This is called an FI curve. And so the idea of a firing rate model is to not bother trying to model the details of all the spikes, but just capture changes in the firing rate itself, which from the perspective of neural coding is also thought to be what matters most about the function of the system. More specifically, we're going to focus a lot, especially at the beginning of the talk, on what I'm going to call recurrent firing rate networks. So here I have two populations of neurons. One here in blue, these are the Y population. And then up here in orange is the X population. And I'm going to model the dynamics of all the neurons within this recurrently connected population with a simple differential equation where you have a tau dy dt minus yi. So that means in the absence of this input term here, it would just exponentially relax to 0 with time constant tau. But then you drive it with some input current, which is computed by summing over all the presynaptic neurons from either the recurrent population or the input population through some weights W, and then it passes through something like an FI curve to convert that input current into a firing rate. Now, the first thing I want you to notice about this equation is I'm going to focus a lot on steady states. So what are the kind of steady state properties of networks? And once I set the time derivative equal to 0, I get these algebraic equations relating, for example, the firing rate of neuron I to the firing rates of neurons Y, all the others that are providing input, as well as all the input neurons. And for notational convenience here, so I don't have to distinguish when I'm talking about W, I'm going to use this weight matrix W to kind of collect inputs from both the recurrent population and the input population. So it's a rectangular matrix where the number of rows is the number of neurons in the recurrent population, which I'm calling D, and the number of columns is D plus I, which includes also the number of input neurons. Now, I want you to notice that once things have reached the steady state, effectively you end up with what's called a feed-forward network, where the activity of Y is just some nonlinear function applied to the weighted sum of its inputs. And as almost all of you surely know by now, this type of architecture is fundamental in terms of how AI and modern machine learning work. Okay? So this is the class in general of neural networks I'm going to be dealing with, and I'm going to kind of have more specific versions of this in several different biological settings. Now, what I want to do is talk about dissolution space geometry. Because going back to the motivation, I don't want to just find a single model that works. I want to understand what is the space of models that work. And I think this is easiest to illustrate with a very simple toy model. And so what I want you to do is look at this example, which is the simplest thing you could probably imagine. You just have one neuron Y that's being driven by two input neurons, X1 and X2, through weights W1 and W2. Now, I want to specify the ensemble of synaptic weights that are consistent with some set of data. So before I get any data, I'm just going to make a first assumption, which is that synaptic weights are not infinite. So what I mean by that is that if I take the norm of the weights, the sum of W1 squared plus W2 squared, that's bounded above by something, capital W squared. Now, the idea is that once you start to make observations on the system, you start to get some constraints. So for instance, let's imagine that we know that if the input was 1, 1, the output is 1. Then the target neuron responds. And what that means is that W1 times X1 plus W2 times X2 is equal to Y. This is because for a lot of the talking, we'd be working with FI curves that are threshold linear. And what that means is if their input is below threshold, you get 0 response, and it increases linearly beyond that point. going back to the example above, this just applies an algebraic constraint that W1 plus W2 is equal to 1. So what that does is it takes this initial sphere of possible weights, which I've denoted by deep yellow here, and it reduces it to a line. And that line corresponds to all those weights, W1 and W2, that satisfy this constraint that W1 plus W2 is equal to 1. The next thing I want to point out is, let's imagine you have a second stimulus condition where the neuron doesn't respond. So for example, maybe you have 1 for the input here and -1 here. Because the neuron doesn't respond, you don't know actually that its input current's exactly 0. All you know is it's subthreshold. And what that then does is it translates into an inequality constraint, where W1 times X1 plus W2 times X2 simply has to be non-positive in order to guarantee no response. And what that does then in terms of the geometry on the right is it says W1 minus W2 has to be less than or equal to 0. And in particular, of that line of solutions you had before, half that line disappears. And you now have a scenario where everything that's in deep yellow is consistent with these two activity patterns and this constraint that the weights are non-infinite. So far, so good? Now, the first thing I want to point out is that in this ensemble of candidate weights, there's an infinite number of possibilities because you have a continuum. But nevertheless, that doesn't imply there's not interesting features within that ensemble. And if we look at this particular example, you see that even though there's an infinite number of weights, the value of W2, which is the synaptic strength from the second input neuron onto the target neuron, is always positive. Okay? And therefore, from a biological point of view, if this was a realistic model, then you would predict that there must be a synapse, because all the models within this class that can explain these response properties have to have a positive weight. For the remainder of the talk, I'm going to go through a couple different specific biological examples kind of inspired by this NITMB philosophy, where I'm going to first introduce some sort of biological problem that inspired me as a neuroscientist, that I wanted to understand something about how the structure of a network related to its function. I'm then going to show how, for that class of models, we can set up a solution space. And that was like the simplest toy model, but it'll be kind of more tailored for the specific biology problems we encounter as we go through. And then there'll be a little bit on, I think the new math that is required when you want to kind of do this type of a thing is to understand solution space geometries. And what I'll do is that for each of these biological problems, I'll explain what aspect of the solution space geometry we gain new mathematical results for. And then finally, I'll point out how that mathematical insight into the solution space geometry is directly translated into an impact on the biology. And so I hope that this kind of like illustrates what I take at least to be the exciting premise of the NITMB type model, where you start with biology for inspiration, but that leads to new math that leads to results and leads to improved biology. So the three examples I'm going to go through are going to be, from a biological point of view, quite different. So I'm going to start going back to the zebrafish, where I began to talk by talking about circuits for binocular integration. After that, I'm going to move to the fruit fly, and I'm going to talk about circuits that are involved in maintaining heading direction. And then finally, I'm going to move to the mouse, and I'm going to talk about circuits that are involved in learning and memory. And for all three of these parts, they're going to have that same basic architecture of biology inspiration, solution space setup, math associated with that solution space, and biological impact of that understanding. All right, first part. So this was probably the problem that inspired me to care about ensemble modeling in the first place. And so this goes back to that complicated model I showed on the first slide, sitting between the structural connectivity and dynamics. One part of that model does binocular integration. So the idea here is that we're looking at fluorescence data, again, inside the central brain of the zebrafish. And the first thing you're seeing here is the signals that you observe in the axon, so that's the outputs of the retina, on the left side of the brain and the right side of the brain. So it's very clear that green and purple are segregated, and that corresponds to signals from the left eye and the right eye. And so what this is showing you is that at the level of the inputs, there's a segregation where the signals project exclusively to the contralateral eye. Retina recipient brain area, called the pretectum, extends its dendrites into these retinal arborization fields, and it develops binocular representations. So for example, you can see that because there's many neurons here that are white. That would correspond to responding to motion in either eye. And you see it also develops this lateralized structure, where now all of the responses to leftward motion independently of the eye are on the left side of the pretectum. And this is one example of a map. But what we did is that we went through and we characterized, well, what are the different patterns of response that you get of neurons in this pretectal area? And one part of that model that I described to you is a neural network model of how you could wire retina signals into the pretectum in order to generate, in order to generate the diversity of responses that we needed. And this model class that we set up initially just from the perspective of modeling this biological data set was, again, a threshold linear recurrent neural network where the pretectum has to have recurrent connections because it has to generate binocular representations without getting direct input from the retina itself. It has to do it inside of itself. And also, I'm not showing you the data for this now, but it has approximately threshold linear binocular integration. So there's a hard threshold, but it seems like it sums its inputs monotonically and linearly once you go above the threshold. So now this leads us to a setup that was similar to how I introduced rate networks in the beginning. We're going to kind of ask, given some number of stimulus conditions, so these are patterns that you show to the retina, the neural network will settle into some steady state, which will be the pattern of neural coding in the pretectum. And that'll define, because of the fact that rate dynamics have settled down, some pattern that says how the output of each neuron yi depends upon all the inputs, their activity, and the candidate weights. Because we're going to focus on steady state response patterns, everything's fixed points. And because everything is fixed points, you can actually very rigorously decompose the recurrent equations into a set of feed-forward ones. Because if you're asking about what are the synaptic connections onto this individual neuron, for example, you know all of the presynaptic activities from all those neurons. And this is one of the special features, again, of the zebrafish, is because it's doing whole brain imaging, you can get away with assuming that you're incorporating all the neurons in your model. Because if it doesn't work, then you need to add more neurons. You have access to everything. Okay, so it's an approximation that you make. And then the idea would be that you have these steady state response patterns. So you have some input that occurs at time 0. The firing rates evolve dynamically over time, but eventually they all kind of settle at some value. And what I'm showing you here is that for some individual neuron who are trying to figure out what we can say about synaptic connections onto it, we have p different steady state values of where that neuron goes given different input conditions. And because it's a threshold linear neural network, and again, this agrees with the data, all of these are going to either be positive or 0. Okay? So for each neuron, we have p response patterns that are constraining what we can say about the synaptic inputs. In terms of the synaptic inputs, we have this firing rate equation, and we have n incoming synapses. So we have n unknowns and p response patterns. Now, when you're working in the regime of whole brain imaging and very large data sets, you're actually in this undersampled regime where the number of patterns is less than or equal to the number of neurons in the system. And I'll show you how, in this case, it actually allows us to get an analytical characterization of all the feed-forward and recurrent weights that can generate these fixed points and allows us to do a geometrical analysis of the associated solution space. All right, so let's go through this math briefly. So the first thing I want to do is I just want to define a n by n full rank matrix, capital X. And the way that I'm going to do that is that for the first p rows, I'm going to set those equal to the steady state response patterns of the inputs. Okay? Now, because p is less than or equal to n, there's generally going to be n minus p missing rows. And what we're going to do is we're just going to extend X to a full rank matrix in an arbitrary way as long as it ends up to be full rank n. So this, you can think about the n minus p remaining rows just being a basis for the kernel. In this coordinate system, it turns out that everything gets very simple. And so why I say it's a coordinate system is you can write down all the steady state equations very simply as y mu equals f of eta mu, where eta mu is a linear combination of the w's that depend on x. And you can think about x as representing a basis transformation matrix, that it's taking your n-dimensional space of synaptic weights, and it's writing them in an alternate coordinate system that has made the constraint equations very simple, that everything is just y mu is equal to f of eta mu. So what ends up happening when you do that is that the solution space has very natural coordinates that are very easy to specify. So there are 3 types of dimensions. So the first thing that happens is imagine that y mu was positive. So if y mu is positive, that means eta mu has to be positive, which means that f of eta mu is just eta mu. And so you end up with an equality constraint that just says y mu is equal to eta mu. So you have one dimension in this high dimensional space that's just completely specified. If y mu is equal to 0, you can't say that it's equal to 0, because again, the same argument I showed you in that toy model, all you know is its subthreshold. And what that then does is it provides an inequality constraint, and we call that therefore semi-constrained. And then finally, for all of these coordinates where mu is bigger than p, to remind you, these are just an arbitrary basis put on the kernel. These are completely unconstrained. You can't say anything about them. There's just unknown coordinates that are coming from p being less than or equal to n. And so from a geometrical point of view, what the solution space looks like is that there are certain coordinates that are constrained, where the solution space is a fixed distance in those directions. There's certain directions called semi-constrained, where you have these half-plane type structures. And then there's certain constraints, dimensions like these unconstrained, where everything is completely flexible. And once you have this parameterization of the solution space, this is the exact description that works for the recurrent problem too, because we can do this neuron by neuron, because we know the steady state levels of everybody, then you can just go back to the physically meaningful synaptic weights by applying the inverse basis transformation to any solution you specified in eta coordinates. Okay? And then therefore what that looks like is you've just kind of done a linear transformation on this space to get it back into the synaptic weights. So this construction allows us to go back to this binocular integration problem I described earlier and ask some questions that we think are quite biologically interesting. And so let's just imagine we want to know what is the minimal synaptic connectivity that's required to generate these responses. What we can do is that we can specify now for any set of activity patterns where p is less than or equal to n, what is the geometry of the solution space? That's what's cartooned now by this deep yellow wedge now in this kind of higher dimensional sphere. We can also define a plane in this neural network space, which is just the value for which an individual synapse is exactly equal to 0. So that's just a plane. And therefore, in a scenario like this, where there's no intersection between the plane where an individual weight's equal to 0 and the solution space shown here in deep yellow, then this corresponds to a scenario like I showed you on the first slide, where even though there's a huge number of solutions, all of them have the same sign, because all of them are on the same side of the separating hyperplane. And then therefore, this would allow us to predict that there must exist a synaptic connection between these neurons from the entire class of models, rather than just from an individual model. And it turns out that for this problem, the solution space description I provided to you is complete for this kind of p less than or equal n condition. And between these two papers, we explain how you can solve this geometrical problem of assessing when there is an intersection and when there's not. We have, for the case of orthogonal patterns, in this first paper, we have an exact closed form solution for that. And then in this follow-up work, we have a very close to exact but approximate analytical solution for that, as well as exact numerical algorithms for finding whether there's a solution. Yeah.
How much does this depend on the fact that your constraint is spheres, where basically you're now having combinations of synaptic inputs having some constraint? What if I just wanted to say an individual synapse has an upper bound, and now I live in a world of cubes instead of spheres?
Yes.
How much does this change?
Yeah, so we've done a variety of analyses like that. We don't have the full analytical characterization for that. And the reason is that this sphere type thing corresponds to an L2 constraint, and that allows us to map everything to quadratic programming problems. So I think that we do have numerical results where we kind of consider other types of constraints, but we don't have we also haven't worked as hard on those other versions. But the analytical results are for the spherical constraint. But you can imagine putting a big enough sphere that it just kind of contains your desired cube. And then, if it's consistent within that sphere, of course, it's consistent within the cube too. All right, so we can do this now for a real biological data set for each and every synapse in the system. And again, it's very numerically efficient because the solution space is analytical, and we have analytical formulas that very well approximate everything. And we are now collaborating with experimenters who have actually done exactly the type of experiment you would want to test this, where what they've done is that they've done functional imaging of these neurons in the pretectum to measure their response properties. Then for the very same animal, they've sacrificed it and measured synapse resolution connectivity, and they have everything aligned. And so they're actually then able to test our theoretical predictions with existing data, just with additional data analyses. And we can then set up within our modeling framework, just like I kind of started at the beginning, we have some retina inputs that are coming from the eye, driving this recurrent neural network of pretectal neurons. This is a representation of, don't worry about the details, but what the response patterns look like for this particular experiment. And we can evaluate what are the strongest predictions. And the way that we do this more specifically is that the scenario I show you here where there's no intersection is for a fixed value of W. And if you imagine that you made W bigger, eventually you're going to get an intersection. And from a biological point of view, we don't know what that capital W constraint would be. And so what we actually do to kind of make these predictions is we figure out, well, what is the largest sphere for which there is no intersection? And we can evaluate that number for each and every synapse, and therefore create a rank-ordered list of how large of a solution space you have consistency over for every synapse in the system. All right, so that was an example where we're using zebrafish, and we're focusing on making use of the fact we have dense activity recordings in the system to try to make predictions regarding the circuit level synapses. Another version of it would be, what if you had the connections already, but you don't have, sorry, I didn't mean to go forward yet. What if you have the connections already, but you have a less clear functional characterization of the system, but maybe you do have some high-level insight as to what you think the system is going to do? And so that leads us to something called the head direction system in the fruit fly. So there's 4 things going on here at once, but I think it's worth seeing everything at the same time. That's why I don't build it up slowly. So the first thing maybe to just draw your attention to is this thing here. So that's a fly. He's head embedded, and he's walking and turning on a wall. What you're seeing up here is a non-neuron resolution measurement of activity in a circuit. Then you can see that basically what it looks like is a bump moving around on a circle. What you're seeing here is, for that circle, what is the synapse resolution anatomy structure. And you can see that there are 8 different colors. These correspond to 8 different neurons, very neatly tiling this circle, into 8 compartments. So the way that this system is thought to work is that through these patterns of synaptic connections, it generates a bump of activity that encodes the heading direction of the animal, essentially like it's a compass. And you can see that directly here. So here what you're seeing in blue is what the animal's actual rotation is. And what you're seeing in red is what you would think it was if you decoded this as basically just a bump of activity whose location on a circle corresponds to the head direction of the animal. So it is literally a compass sitting in the middle of the fly's brain. It's a circle where the neural activity points in the direction the fly is heading. It's ridiculous, but it's true, okay? And what you're seeing here is that that's accomplished by a very small number of neurons. So here there are 8 different computational units that are somehow managing to represent the heading direction very precisely. And moreover, it seems as though the representation you would get of that head direction system is much more fine than you would naively assume if you just thought that each one of these neurons had a preferred angle and if that one's the active one. So the question we have here from a biological point of view is, okay, can we understand how you could get exact ring attractor dynamics? That's what this is called in a computational neuroscience sense, where you have a continuum of fixed points along a ring, in a way that's consistent with the synapse resolution structure in the connectome. And what would be the predictions, for example, in terms of the activity that would underlie that, which you don't directly measure here because everything is very blurred out because of the nature of the neural signal. The way we're going to do this is that each one of these gray circles would represent one of those 8 computational units. And I've now introduced also these black units. And that's because all of these gray guys are excitatory neurons. And it's known that to create and maintain this bump, you also need inhibition. So I didn't show you the anatomy data, but it's also, there's a population of 8 inhibitory neurons called delta 7s that are thought to mediate this inhibitory profile. Now the idea is going to be that we want to say, okay, well, given some pattern of connectivity involving these computational units of excitatory and inhibitory neurons, is it a ring attractor or not? The way we're going to address this is by drawing a mapping from this 16-dimensional model, well, 16 neuron model, to a reduced model of just effective neurons that combine the excitation inhibition into one. And it turns out that this circuit, both at the level of the anatomy and in terms of the function of the bump, seems very symmetrical. So we're additionally going to make this assumption that even though you have these 8 different computational units, that if you rotate the whole system by 2 pi over 8, the parameters stay the same. So it's rotationally symmetric. So if this initial model is rotationally symmetric, what that implies in terms of this effective model is that to compute the effective weights between 2 excitatory neurons, you have to sum over paths involving the inhibitory neurons. And if it's the case that all the inhibitory neurons are active for every location, So what I'm showing you here as well is in brown, I'm showing you which are the neurons that are active for some given location, and what are the neurons that are inactive, here shown in unfilled. And so if it's the case that the inhibitory neurons are always active, we just don't know this from the data, it turns out that the rotational symmetry here translates directly into a rotational symmetry here, and you can parameterize the whole thing by a few parameters, as I'll get to in a second. Well, sorry, right here. So there's only 3 parameters in that rotationally symmetric regime. So you've got some W1 about how strongly you synapse onto your nearest neighbor, W2 is your next nearest neighbor, W3 the next nearest, and then there's also a long-range W4 connection that would go from one of the active neurons to the inactive neurons. Now the other thing that can happen is that if it's the case that some of the neurons in the inhibitory set are inactive for a given location of a bump, you actually break symmetry. And the reason is that you have a path from this neuron to this neuron by activating that inhibitory neuron and getting a back synapse, but you don't have the reverse path because that neuron is inactive. And it turns out that what that translates to mathematically is that you have a mirror symmetric effective ring attractor network. And so you end up with now more parameters where rather than just having 3 involved in maintaining the bump and 1 not maintaining, you end up with 6. You've got is that right? Do you get 6? No, you get 8. Sorry, I'm just counting again. Yeah, you get 6. So you have the number of local ways of arranging it while maintaining mirror symmetry. So far, so good? So what we now want to do in terms of the ensemble modeling point of view is we want to ask, okay, well, what do we need to get exact ring attractor dynamics in terms of this parameterization for the symmetric model and this parameterization for the asymmetric model? Long story short, it again turns out we can analytically solve this problem. And here I'm showing you the complete solution space for the symmetrical model. So it turns out that this most local synaptic connection, W1, has to be positive, and it can only take the values between 0.5 and 1 in order to maintain a continuous attractor. Once you specify W1, W2 is fixed as 1 minus W1 squared. That's one of the 2 curves you see up here. And W3 is fixed as W1 times 4W1 squared minus 3, which is the other curve you see up here. And then finally, in terms of this long-range parameter, you don't get any equality constraints associated with it because all it's doing is maintaining the neuron below threshold. So it's like a semi-constrained dimension, if you will, in the way I set things up in the previous talk. And then that, therefore, just gives this envelope here shown in light blue, and anything less than that's a solution. So in this particular representation, any value of W1 that has the blue line there, the purple line there, and the light blue anywhere below that is a valid solution. In terms of the mirror symmetric model, we can again get an analytical solution, but now it's higher dimensional, so it's harder to represent. But basically, the main qualitative features is that the basic dependencies of things look very similar. The biggest difference is that you get essentially no constraints because on the longer range connections. So now we have a description of the exact description of what are the parameterizations in these symmetric classes of models that can support a ring attractor. So what's the biological impact? So now we can go back and ask, is it actually the case that the fruit fly brain is consistent with one of these models? And so this is what the data would actually look like. So these are synapse counts between these EPGs would be those excitatory neurons I mentioned. These delta 7s would be the inhibitory neurons. And we have from experimental measurements what these synapse counts are. Now, to remind you, we have to sum over paths. But synapse counts are not equivalent to weights. So one obvious way in which synapse counts are not equivalent to weights is that if you have an inhibitory neuron, it's going to have a positive number of counts. So you need to be able to rescale to be negative. And generally, there's going to be some per scale factor, at least, that's going to kind of represent how synapse counts translate into synaptic weights. So what we're going to do is we're going to, here I'm showing you every single neuron, but here I'm kind of coarse graining it according to this effective unit model that I showed you as a good description of the systems organization. And then we have to rescale in a synapse type by synapse type way the synapse counts to get synaptic weights. And we're going to use the ensemble modeling by asking whether once you sum over paths in this now neural network model that's consistent with the connectome, you can map exactly onto one of those elements of the solution space I showed you on the previous slide. And the cool thing is that we can. And not only can we can actually do it in multiple ways. And so here I'm showing you what is the equivalent model that we find. We find one of them that's symmetric, which would predict that all these delta 7 neurons would be active, which we don't know, that could be true. And then we get two mirror symmetric solutions that exactly maintain ring attractor dynamics, but are consistent with the connectome, but make different predictions in terms of the activity within the system. And from a high-level point of view, I just want to stop and point out one thing I think is really interesting about this, is that we started with the connectome, but we had to have still some free parameters, because we had to figure out how synaptic weights relate to counts. And we did that here in the way that's commonly done, at least currently, by people who build connectome-constrained models. You introduce a scale factor that converts synapse counts to weights. And it turns out that we could do it, but we could do it in multiple ways. And so this raises the question of how special was the real connectome? Like, could we have done this with another connectome? And it's quite interesting that we find that actually you can move quite a bit away from the real connectome and continue to be able to get exact ring attractor dynamics by rescaling these scale factors. And so there's this concept in computational neuroscience of fine-tuning, that when you have these kind of continuous attractor models, they're problematic because they require synaptic weights to be very finely configured. But here, you actually don't have to fine-tune anymore the detailed pattern of synaptic connections. You just have to keep tuned correctly the scale factors. And that could be achieved, for example, by something like neuromodulation in a biologically plausible way, we think. Also, as it relates to the specialness of the connectome, I don't have anything to show you about this here, just for the purposes of time. But we still think that even though we can do this with many different starting models, there is something special about the connectome, because we've never found another starting point that's far from the connectome where you can get three distinct solutions. It seems like the connectome is in some space where there's really a lot of opportunity nearby compared to other connectivity matrices that we've explored. For the final part of the talk, I want to move into memory and mouse. So the experimental study that got me interested in this at first was during my early days as a neuroscientist, as a grad student, in fact, I had been collaborating with experimenters who were imaging the dynamics of synapses within the hippocampus. So the hippocampus is a region of the brain that's intimately related to memory. It's thought to be where memories are initially stored, and it's thought to be a very rapid, one-shot type learning mechanism that can store detailed information about everyday events. And what we found in this experimental study, when we looked to see over a time period of about 3 weeks, some of the experiments were longer, and we just tracked individual synapses. So these little bulbous things you're seeing in these images correspond to the locus of synaptic connections between neurons. What we found is that some of them persisted, some of them disappeared, and other ones appeared. And there seemed to be a lot of dynamics. That much was expected. But what was surprising to us is if we actually statistically modeled, well, what would be a kinetic model that would account for these dynamics? What we found is that actually the models that best fit the data had no stable spines at all over long timescales. And so although on a finite timescale and with finite optical resolution, you may see some stability here, over very long timescales, it seemed as though everything was flushing and moving away. And that was different than what we found when we applied the exact same statistical inference method to neocortical data, where we found a quite large population of synapses that seemed to persist throughout the lifetime of the animal. So this impressed us and surprised us, because this was more instability within the system than we thought to see. Around the same time, in the same lab, my colleagues were studying how neural codes in the hippocampus, so again, same brain region, change over time. And I want to just introduce you quickly to a concept of a place cell. And so one thing, for example, that hippocampal neurons code for is if you have an animal that's just running back and forth along a linear track, what you find is that there are neurons that code for where the animal is in that linear track. So these cells are sorted so that there's a peak of activity that tiles the position in the environment. And they're called place cells, therefore, because their activity encodes the place of the animal in the environment. Now, when my colleagues now looked at the activity data in a similar type of experiment, they too found a surprising degree of instability. What they found was that many neurons that seemed to be coding for position on here day 5 were dropping out of the representation as time went on. But it wasn't as though that information was lost, because at the very same time, other cells were starting to code for this position. And so the statistics of the representation, the way that space was represented seemed to be preserved, but the detailed cell-by-cell membership in this representation was just constantly changing. And from a theoretical point of view, this underscores the flexibility of a system like this, But it also led me to ask, from the perspective of a memory system, why might you set things up in this way? Like, why have so much instability if one of the goals of the system is to retain information? So now I want to set up another neural network architecture, and I'm going to use solution space modeling again to kind of try to address that question about what you potentially gain by having the system moving around among different possible representations. So the way that we're going to set this up is that we're going to imagine that there's some population of neurons. These are going to be the neurons whose activity changes over time. We're going to call that drift. And they're getting input from some other population of neurons that are inputs through some weights that are going to change. So the weights changing here are kind of emulating those synaptic dynamics part that I started with. And then the representations changing here are the downstream effect of that. The reason I've broken that neuron into two, as you'll see on the next slide, is we're going to kind of talk about the input current that's driving that neuron, as well as what is the activity that results from the FI curve. And unlike the last talk, the last part of the talk, I'm going to assume that there's both a lower threshold and an upper saturation threshold to prevent neural activity in this model from getting too high, because the neural activity is going to change as the weights are explored. Now, what I'm going to do to kind of constrain the solution space is I'm going to say that, OK, although I can change the weights here and change the activity level in the representation, I'm going to force an assumption that there's some low-dimensional projection of that activity that stays consistent, and that would correspond to what is cognitively or behaviorally relevant. So for example, maybe there's a downstream neuron who reads out the activity in this population, and as long as that readout is consistent, you can let the representation drift, and that provides the flexibility you see. I do want to emphasize that I want you to think about these as biological synaptic weights that are kind of connecting inputs to the neurons we're modeling. But I want you to think about this as just a mathematical abstract relationship, because although there's evidence that there's stable structure within these drifting representations, there's no evidence that that structure is linear. And so think about this as some sort of abstract readout, but for now we're going to fix it to be linear for the purposes of getting off the road. So now the way we're going to use ensemble modeling is, it turns out, and you'll see this on the next slide, that we can generalize the solution space picture that I introduced at the beginning by no longer enforcing that all the neurons have the same values, just that the readout stays the same. And then what we're going to hypothesize is that what representational drift corresponds to is a random diffusion within the solution space. So of all the things that can work, what would happen if the system just randomly moved around within that space? And we're going to, again, ask this question of, does anything useful actually happen from this, of just kind of allowing random exploration? So how do we mathematically describe this solution space? So what I'm going to do is I'm going to break it up into several sequences of transformations. So you have your input representation here that gets transformed through feed-forward weights into an input current. That's what's called UX. That then passes through a FI curve to generate the firing rates in the population of neurons that are drifting. And then finally, there's a readout W. And we maintain a consistent mapping from here to here for some fixed number of input-output patterns and allow everything else to drift. W, by the way, going back again to the concept that this W are not weights, this is some sort of readout, we're assuming that there is a stable perceptual structure, which is, again, consistent with the data, but it wouldn't be necessarily linear. And so all the drifting is kind of not affecting what's going on in these abstract W weights, which are specifying the decoding, not synapses in the system. So the way we're going to characterize the solution space is basically just do the same thing we did in that earlier work I described. We're going to characterize it locally in this eta coordinate system that makes everything clean and easy to understand. And then we're going to hypothesize that we're going to generate drift by randomly moving these coordinates. And then we can always just go back to the original coordinates with that inverse basis transformation I talked about before. So the first type of flexibility that we get are these unconstrained dimensions that I introduced you to. These are dimensions in synaptic weight space where there's no constraints at all. And in those dimensions, the neuron just kind of randomly, the weights just kind of randomly diffuse. The next type of dimensions come from the nonlinearities in the system. And so I explained to you about how you got these so-called semi-constrained dimensions from inactive neurons because all you knew was subthreshold. So that persists, but now you get a new type of semi-constrained dimension for when things are saturated, because it now becomes possible that if you're far above threshold, you also have a lot of flexibility because all you know is that you're above threshold saturation. Then the final thing that can happen that's new about this version of the model compared to the previous model is that you can actually change the representation itself just so long as the output doesn't change. And what that means is that for all those engaged neurons, so the engaged neurons are the ones that are in between the inactive state and the saturated state and whose activity levels can actually change. For all those engaged neurons, you can change their activity as long as you stay within the kernel of this readout matrix W. And if we then use this solution space geometry, which is now in a mathematical sense a manifold, because what we've done is that for any starting point, we've specified all the local changes you can make. And so when we generate the diffusion, you're diffusing on this manifold, but we've specified it locally just in terms of the changes that can be made. And when we generate this diffusion, we can get very different starting weights from final weights. And we do it in such a way, which again, by mathematical construction, the outputs don't change at all. Okay? So now we have a mathematical description of the solution space manifold that doesn't distort these input-output mappings. What do we now learn about biology going back again? So when we simulate this system, we found a very interesting phenomena that we did not anticipate, but it turns out this has actually been seen experimentally. What happens is that even though at the time of learning there's a lot of both engaged and disengaged neurons, as the time of drift occurs, the number of disengaged neurons goes up, and the number of engaged neurons go down. So in simpler terms, most of the neurons are becoming either inactive or saturated as a result of representational drift. We think of this as an entropic effect. So again, this thing is diffusing just in a solution space which is bounded by a sphere, and most of the volume of spheres live near the surface. So from this diffusion, you're going to tend to push yourself towards the surface of the boundary. And there, because you have large weights, it's likely you're going to either push yourself above or below threshold. You're unlikely to have a small value of input that leaves you between the two thresholds. And as a consequence, just from the geometry of the solution space, we have a larger number of sparsely engaged representations. And then this then becomes an entropic effect, whereby as you start to explore the solution space, you just see the appearance of many, many disengaged neurons. And it's not as though, going back again to the biological motivation, everything keeps changing. It doesn't stop. It's just the statistics stabilize to a level that's kind of consistent with the solution space geometry. What is this good for? We think what this is good for is noise robustness. So if you imagine, for example, that you had a neuron whose activity level was in one of these disengaged regimes, even if you perturb the input to it, either through changing the input weights or by changing the value of the stimulus that drives it, because it stays, in this case, below threshold, or in this case, above threshold, it doesn't actually change the activity level. And because it doesn't change the activity level, this type of noise won't actually degrade the memory. And it makes a lot of sense that in a learning system, this type of noise, if you will, is going to happen all the time. Because once you've learned this information, you keep learning new information. And as you learn new information, the weights keep changing. Okay? And this is saying that because your drifting is driving you into these sparsely engaged representations, that's having a less detrimental effect on the readout of the system. But I also, and then this is just quantifying that intuition actually holds in the simulation. So if you kind of go back again to these simulated drift simulations I showed you, and you look at what is the sign of the error in a sign sense, if you inject noise into the system, what you see is that the magnitude of the error decreases as a consequence of drift. But we also think this is important. Then why wouldn't you just do this in the first place? If these sparse solutions are so good, why not just start with them? And we think that even though they're very good for memory maintenance, they're actually very hard to learn initially. Because although the exact learning algorithm that biology uses is unknown, in order to learn, essentially by definition, your performance has to get better over time. And if your performance is getting better over time, there has to be gradients nearby. And so we think that from the perspective of learning, you're biased towards having these densely engaged representations, where you could actually manage to change your weights in a way that changed the output and enable learning in the first place. And so put together then, we think about drift as being something that once it coexists with learning can help you find noise-robust representations. And I should comment that this particular project, which I obviously have gone through very quickly right now, my student Manasa is here at the meeting, and she's going to have a poster about that outside. So if anybody wants to learn much, 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 much more about this last part, just go to her poster, and you can learn more. And with that, I'd like to stop and acknowledge everybody involved in this work. Tirtho is a very, very, very good theoretical physicist who's a research professor associated with the lab. He was the first one who kind of recognized this geometrical insight of these constrained and semi-constrained dimensions, which really, I think, you will have seen played a fundamental role in many parts. And he'll be looking for independent positions. And so if anybody's interested to hire an excellent theorist, I'm very happy to connect you with him. Salim is a grad student in the lab, so he's involved in doing the comparison of the EM resolution synaptic connectivity data to the predictions we're making from animals. Manasa Natarajan is here. I already advertised her. She has a poster. She works on learning and memory. And then Lambus was an undergrad in the lab who's now a grad student at Harvard who also contributed to the mathematical analysis of these systems. And then finally, I want to acknowledge the work I presented here involved directly Angel and Sandro from Genelia, which is where I just joined Northwestern back in January 2024. And before then, I was a group leader at Genelia. So this project started then. And funding from Genelia as well as the NITMB, which is supporting this work. Thank you.
Non-sensing zebrafish where there's a lot of sort of background drift and activity in the brain. So how certain are you that the data that you're seeing when the zebrafish are sensing is actually a steady state?
Yeah, that's a great question. So that very first slide where I showed whole brain data and whole brain connectivity, I showed a whole brain model where there's that's a minimal circuit that can kind of bring you from input to output. I was only focusing on a very small part of that in the second part where I talked about binocular integration. And in that part of the brain, it's a retina recipient area where fixed point dynamics are actually a very good approximation. So when you turn on the visual stimulus, the neurons in that region just approach a steady state, and then we're using that steady state. So it's definitely the case, going back to, I motivated this as, this is a biology question I'm interested in, building network models, but we need new math. I definitely have not specified all the solution space geometries we would need to understand everything going on in the fish brain. And so there's lots of dynamics, even within the context of this behavior, would not be captured by this. But in terms of the binocular integration, I think that the formalism we put together is a good one for it.
So without stimulus, that would just be completely off?
Correct. Yes. And then without stimulus, the animal will still behave. Those neurons will not be active. And then when those neurons turn on, they modulate the behavior from spontaneous. Yeah. Yeah. Yes.
And about the last part. Mm-hmm. So you were looking at what happens if that output function is the same.
Yeah.
If you relax that, how much do things change? Is that it, or is that an interesting question?
Yeah, I think that is an interesting question because it could be that the cognitive readout itself is changing. Yeah. So let me kind of address it maybe in three parts. So the first part is what is most complete. So we've done a version of the solution space where you can change U and W, so they can both diffuse. That is not a complete solution. We've only dealt with the scenario where they change independently of each other, because the coordinates about how to kind of coordinate those two things remains an open question. So that's part two. So we don't know how to coordinate those two different processes, but also if one of them is this abstract cognitive thing and the other is this low-level thing, it's not clear they could be coordinated in a biological point of view anyway. But from a mathematical point of view, there's still progress to be made there about how to kind of coordinate changes across multiple layers. And then another thing is, I mentioned here that NITMB is now funding this project. And so the funded extension to this is that we are analyzing now nonlinear readout structures, because this seems to be a better description of the biological data. And so we're collaborating with Andrew Fink's lab, who studies representational drift in puriform cortex. We're using that data to quantify what drifts and doesn't drift, to kind of quantify what is the stable manifold, and then revisiting all these things with a more biologically realistic model about how things are changing.
Of memory is because of you have a bond there instead of having like a inhibitory neuron. So what will happen if you don't have that like bond, but instead you're putting like inhibitory neuron in it?
Right, that's a good question. So we do, so another thing I didn't mention earlier that's another simplifying assumption of all of this is that synapses in these neurons, some can be positive, some can be negative. So we don't enforce the distinction between excitatory and inhibitory neurons. So synapses can be, mixed signs. So with regard to this question about do we need that upper saturation, so all the examples I showed here, I just arbitrarily set the inactivation level at 0. And if you set the inactivation level at 0, then it turns out that you're very likely to end up with large value. Even if you have just large random weights mixing excitation inhibition, there's a high chance that some of them are going to be really pushing neurons very, very high. And so what ends up happening is the neurons get very, very active because of these high weights. But we can avoid that effect by introducing a lower, adjusting the lower threshold so that a greater fraction of the solution space only uses the inactive semi-constrained dimensions. And that's also generates obviously more realistic patterns of drift, because in biology we don't think as though the system's actually reaching saturation. But from the viewpoint of illustrating the concept simply, we just kind of use this minimal model. But that one where you kind of introduce a bias, so you actually need a positive amount of input to even activate the neuron, can give you all the stuff we just described without needing the saturation.
I see. So in principle, if you switch the threshold linear function with the upper bound with something like a tangent edge function, you would still have the result that you have a stable memory preserved against noise.
Yeah, and also in that very first paper, you can relax this threshold linear thing, and now we have threshold linear threshold. That's actually quite relaxable.
Okay.
So there's a way to transform the data you're using to constrain the model based on just, the intuition is you just take the inverse of that monotonic nonlinearity and you can map it to something that's threshold linear.
Okay.
And so, we can handle those scenarios too.
Thanks.
Maybe I'll stay on this side for a second, yeah.
I don't know if it works or not. Okay, so just piggybacking on that questions, and also because all of the data, large scale data you're doing is calcium imaging, right? So the dynamic range of your FI curve is limited by the threshold detection and the saturations. So if you have actual spike timing, for example, if you can somehow record 1,000 neurons at the same times, does that change your solution space?
Yeah, so that's a good question. So for the first part, where we were using real activity patterns from pretectum, we haven't seen any evidence of saturation there, because it really looks like when you kind of look at how it integrates its inputs, it seems to be linear. So it seems to be very precisely integrating its retinal inputs linearly. So we think there's not a big saturation effect there. And then that is the only one where I actually really used activity data directly to constrain the system. To your question more generally, for the drift thing now, we actually are working with spiking data. So Andrew Fink doesn't do calcium imaging. He's doing neuropixels recordings. So we'll be able to kind of look and see to what extent these types of things can be applied. We've also done simulations. One of the things I like about this ensemble modeling approach is clearly I'm making lots of biological assumptions that are not correct. Neuron spike, right? But what we can do is we can make predictions with this based on the FI curves and see to what extent it can accurately predict spiking data. Because just because neurons spike doesn't mean you need to account for that to kind of predict the synaptic structure. And so we've done a variety of simulations to show that it's relatively robust to model mismatch issues too, because of the fact that, again, you're analyzing the whole solution space. And when you find consistency so broadly, we intuitively think, and it seems to empirically be the case too, that it therefore is also less dependent on the fine-scale details of how you set up the problem. Yeah.
I found very particularly interesting the final part where you said that sparsely engaged solutions are much more robust to noise, and then you proved that. And I was wondering if, it's a relatively easy experimental task, right? Have you tried to see if animals actually, once they learn the task, if you keep, they learn the task and then you perturb the stimulus with noise, do you think that the robustness for the robustness to this noise is going to be stronger at the end?
Yeah. Yeah. So I have. I'll answer that again in 3 parts. So the first part is that this high level of engagement followed by sparsification has in fact been observed experimentally in the hippocampus, I think, right? Hippocampus. But that came after we developed the theory. But we are of course very excited to see that. So that isn't a real empirical finding. The concept that representations are denser at the time of encoding was already out there, but to see a kind of real curve was new. So that's one part. The other thing that's an issue, right, is to remember I showed you at the beginning the statistics stayed the same. And so most of the experiments are kind of living in that part of the curve where the sparseness isn't changing. And then therefore the robustness also isn't changing. So it's not like you need, the reason you have constant drift is because you're constantly adding new memories, need to keep on maintaining them, noise robust, but you reach a noise robust level for any given memory quite quickly, which again, I think is one of the benefits of this, because it's just an entropic feature of the solution space. It's not like you need to get to a specific point, you just need to get away from where you started, and then you're gonna be okay. And then the final part is that we actually do have a experimental collaboration, again involving my student Manasa, where we've looked at a learning task that takes about a week for the animal to actually acquire the task performance. And we've been using that data set to try to look at what dynamics are attributable to drift and to learning. And coincidentally, it was actually published today, so you can look at it online. It was already online, but it's easy to find because it's in today's press. Yeah, go ahead.
In the last part of your talk, you formulated a nice sort of hypothesis that it's, and proven it, that it's, for learning you need to be in a linear regime or in a responsive regime, but for robustness you need to kind of move towards iteration. So how much do you think it applies to systems other than neuronal networks? In particular, think of gene regulatory networks in a cell or artificial neural networks. Have you thought about it, and maybe just to make it a little bit more precise, what people expect for gene regulatory networks or any other molecular networks is that any weight without selection will go down in time because evolution lose it or use it or lose it.
Yeah.
And your entropic argument, does it rely on this, that if nothing is, if the weight is not selected, it just goes down in time?
Yeah.
Or because I did not completely understand your entropic argument, that's why I'm asking.
Yeah, no, okay, so let me, I got excited by your first question, so I'm not sure if I fully heard your second one, so if I don't answer it, just follow up. So have we thought about gene regulatory networks? The short answer to that is very minimally, and we'd be interested to think about it more. So I presented this work to a group that included Michael Elowitz in the audience, and he came up because he's building these synthetic networks about could we use some of these solution space methods to kind of understand the space of gene regulatory networks that can achieve certain types of dynamical phenomena. And we've had a few conversations about that, but haven't really followed up deeply. And that was actually, we didn't really talk about it from the perspective of robustness. And so I'd be very excited to chat more later in this meeting about what specifically you have in mind. And then,
yeah, actually, Elowitz is a perfect example because this dimerization networks is exactly what I had in mind, and we were working on them for a long time, and I know exactly what Michael did recently. I chatted with him.
Yeah, exactly. So Michael and I have had a few meetings about this to see if we could generalize the math to work for gene regulatory networks and specify solution spaces there. And the honest answer is that we started to do it, it was interesting, but that's exactly when I moved, and so it kind of fell off the radar. So.
Okay. Thank you.
I had a question on the ring neurons part. When you're rotating the ball and you see the bump of activity go around, is there drift if you say go around like 10 times? Does it go around exactly 10 rotations? And if you go 10 rotations forward and 10 back, do you come back almost to the same point, or does it drift with noise?
Yeah, there of course will be some level of drift. It's pretty stable for relatively long periods of time. So that data that I showed you was an experiment where it was following it for that whole duration of the experiment, which I'm not sure exactly how long it was, but it was on the time scale of minutes. One important thing is that because they don't have, they're walking on a ball. This isn't a realistic scenario, so there was a scale factor that's wrong. So I don't know if you noticed that so it doesn't come back, if it goes 360 in real space, it doesn't necessarily come back to the exact same point because it doesn't know how much it's actually rotated because it doesn't have that calibration in the system. But if you turn on the lights, it can calibrate the system, and it also, if there was drift that occurred, it uses things like visual landmarks to lock you back to where you'd want to be. So the integrating part of it is what it does in the absence of cues, but the fully functioning system also uses other things as anchors. Yeah.
Thank you.
Thank you.